Thanks to the organizers and the Allen Institute for the opportunity to talk here today and tell you about the work going on in my lab where we're using zebrafish to understand heart development with the goal of trying to better understand how congenital heart defects in humans arise. So congenital heart defects are essentially structural malformations that arise during heart development. It's one of the most common birth defects in humans. This slide shows that we already know many, many genes that when mutated can give rise to heart defects in humans. But even after a lot of work, it's thought that we really only understand about 20% of the genetic basis for heart defects. And what makes the genetics of heart defects even more complicated is that it's thought that there's could be a very strong role for modifier genes. And so some of the evidence for this is that mutations in the same gene or even the exact same nucleotide mutation in the same gene can give rise to different severities of heart defects. So for example, this has been shown for the cardiac transcription factor NKX2.5. And so we want to use zebrafish as a model not only to better understand roles for new genes in heart development, but to help understand how these genes interact with each other. And so some of the advantages of zebrafish for studying heart development in particular are that the embryos develop outside the mother and develop very rapidly, so that by 24 hours, the embryos have already formed a muscular heart tube, shown in this cartoon here. And this heart develops from cardiomyocyte precursors that start on lateral sides of the embryo. And these cardiomyocytes are one of the main building blocks of the heart. And then by 48 hours, the embryo has already made its two chambers, the ventricle and the atrium. And in live embryos, we can see these chambers pumping and blood moving through the heart. Some other advantages are we can get hundreds of embryos per day. It's very straightforward to do procedures like genome editing. And so essentially, instead of having cells in a dish, we have our zebrafish embryos in a dish. And so I hope that some of these advantages of the fish model will come through in my talk. In addition, you'll probably see some of the challenges that we have in using zebrafish to model human genetics. I'm going to tell you about two stories today. One is a recently published story where we've used genome editing in fish to characterize the role of a genetic variant in congenital heart defects. And then I'll tell you a more unpublished work about how we're studying roles for Pbx and Meis factors in heart development and their roles in cardiomyocyte differentiation. So what are these Pbx and Meis factors that I'm going to be talking about are both homeodomain transcription factors. And one of the reasons that we've been so interested in Pbx factors in particular is that they've been shown to act as pioneer transcription factors and can bind closed chromatin and then work very closely with the cofactor Meis proteins to recruit other master regulatory transcription factors to help regulate gene expression in a variety of cell types during development. So one particularly well-characterized partner for Pbx and Meis proteins are the Hox proteins. These are well-known, have well-known roles in anterior-posterior patterning and embryonic development. Work from myself and Stephen Tapscott's lab showed that Pbx and Meis act as cofactors and pioneer factors for the MyoD protein and skeletal muscle differentiation. There's also been work showing that Pbx acts as a pioneer factor for the estrogen receptor in breast cancer. And what I'm going to be telling you about today is how we're thinking of our hypothesis is that these Pbx and Meis factors act as pioneer factors for important cardiac transcription factors in promoting myocardial differentiation. And so some of the strongest evidence for this hypothesis comes from genetics. So both zebrafish and mouse models that are mutant for various Pbx genes show defects in heart development. So my lab showed that loss of the Pbx4 gene in zebrafish leads to cardiac defects. So you can see swelling around the heart, some blood pooling in front of the atrium. And I'll tell you a little bit more about these phenotypes in a bit. Work from Krinstan Kunis and Qingping Chang's lab showed that when they take away different alleles of the mouse Pbx 1, 2, or 3 genes, that they could see different severities in heart development, in particular in the outflow tract of the mouse heart. And this was dependent on how many alleles of these Pbx genes they removed. And work from Licia Soleri and Cecilia Moens have showed that these Pbx genes are very broadly expressed and are highly redundant with each other. And so to study Pbx function, it's important to knock out multiple copies of these genes. And one of the advantages of the zebrafish is that only 2 Pbx genes, Pbx2 and Pbx4, are expressed during early zebrafish embryonic development, during early heart development. And so recently, my lab has engineered mutants for the zebrafish Pbx2 gene and combined that with loss of zebrafish Pbx4. I can tell you when we take away zebrafish Pbx2, this MZ refers to removing both the maternal and zygotic contributions of Pbx2. These animals are normal, but when we take away both Pbx2 and Pbx4, we see much more severe defects in heart development. And we think that these defects are arising as an early cardiomyocyte differentiation problem. So these are showing you fluorescent in situs of early zebrafish embryos, showing you those early myocardial patches stained in purple for myel7, an early heart muscle marker. So when we take away both Pbx2 and Pbx4, this early myel7 expression is absent. So that indicates that these are needed to help activate myel7 expression. Myel7 eventually turns on in these animals. And so these are showing you hearts of these animals at 3 days. So now they've formed a ventricle, an atrium, and in green, this marks the outflow tract of the zebrafish heart. So animals that lose Pbx4 do have defects in both the myocardium and the outflow tract, but animals losing both Pbx2 and Pbx4 lack an outflow tract, at least by this marker, and have defects in the development of the myocardium. So we see these strange bulges off of the myocardium that we're still working to characterize. But what this indicates is that the Pbx proteins do have an important role in helping to promote normal heart development. And the work from Maves also emphasized this as well. And so the question has been, until recently, there has been little evidence that these Pbx factors have played a role in congenital heart defects in humans. Except there's a couple papers recently, so work from Ann Slavatnik identified variants in the Pbx1 gene in patients that had heart defects. These patients also showed other developmental abnormalities. And then this group showed that in a set of patients that had isolated heart defects, that they showed different variants in different Pbx genes. Including one particular variant in the human Pbx3 gene. It's a C to T, or alanine to valine change. It was found in a higher frequency of their patients than in controls, and it's predicted to be deleterious to the structure and function of Pbx3. And in particular, this variant is found in a very highly conserved alanine stretch, not only in the Pbx3 gene, but also in other Pbx proteins. And so we thought that we could use genome editing to test whether this alanine to valine change found in human Pbx3 could contribute or be causative of heart defects and could test this function in zebrafish. But we decided not to test this variant in the zebrafish Pbx3 gene because it's not expressed during early heart development, and we knocked it out and it has no function, at least as far as we can tell in early heart development. So we engineered this variant in the conserved site in the zebrafish Pbx4 gene. And we used CRISPR to do this. Many of you are probably familiar with how we can do this in zebrafish. We just inject Cas9, a guide RNA, and an oligo containing the C to T change that we want to introduce in the Pbx4 gene. We then raise fish that have been treated with these CRISPR components, can use a PCR assay to identify fish that are transmitting this mutation in their germline. And then we can sequence and identify fish that contain this exact C to T change or alanine to valine change in the absence of any other mutations in the Pbx4 gene. So we generated these fish and found that fish that were homozygote for carrying this variant looked completely fine as far as we can tell. So adults are viable and show no obvious heart defects. And so we then wanted to ask whether this Pbx3 allele could function as a modifier. And so I told you that in the paper that identified this variant that they did see the presence of this variant in controls. And so that indicates that on its own, this variant is probably not causative of heart defects, but it could enhance the effects of other mutations that these patients could be carrying. And so in zebrafish, we decided to test whether this variant enhanced the effects of a mutation in HAN2. So HAN2 is another important cardiac transcription factor that's been shown to play roles in human heart defects as well as heart development in mice and zebrafish. And so what we did is we crossed fish carrying the A to V allele with fish carrying a HAN2 mutation. And this experiment got more complicated than we initially anticipated, but I'll take you through what we saw. So these are zebrafish embryos showing you in C2s again for our MYL7 myocardial marker. So here is showing you the heart tube in wild type. Zebrafish carrying that A to V allele show a normal heart tube. Zebrafish that have loss of Pbx4 gene show defects in the morphogenesis of the early heart tube, as I showed you previously. Zebrafish that are lacking HAN2 also show defects in the early MYL7 expression and formation of the early heart tube. And what we found is that if in HAN2 null zebrafish mutants, if we add one copy of loss of Pbx4, those fish looked quite similar to the HAN2 null fish. However, if we now have a HAN2 null fish that contains in trans the Pbx-AV allele, in trans with a Pbx4 null allele, now we see enhanced effects on early heart development in these fish. And so the early myocardial precursors have not even started to migrate together into the center of the embryo to start to form the heart tube. And we were able to quantitate the effects of these alleles by measuring the distance between these two domains. And essentially what we showed is that this A to V allele does seem to act as a modifier of HAN2 in zebrafish heart development, which suggests that in humans that this Pbx3 allele could be a potential modifier of congenital heart defects in humans, potentially depending on what other mutations these patients are carrying. And what this also means is that we can make a patient's fish model and test candidate genetic variants, essentially not only in heart defects, but in other diseases as well. And so we recently published this work earlier this fall. And now I want to tell you, we started a little late, right? I want to tell you some unpublished work about how now we're studying roles for Meis factors in myocardial differentiation. So I mentioned before that these Meis proteins are close cofactors with the Pbx proteins. And so in work a few years ago, I showed that Pbx and Meis could bind together on promoters of not only skeletal muscle differentiation genes, but also our heart muscle differentiation gene, MYL7, suggesting that Pbx and Meis are working together to control MYL7 expression. And furthermore, more evidence for Meis, role for Meis in heart development came a few years ago in collaboration with Chuck Murray's lab and working with Christy Stoic-Cooper when she was in Randy Moon's lab. And we showed that in zebrafish, Meis2B is expressed together with an important cardiac factor, Gata4, in very early myocardial precursor cells in zebrafish embryos. And if we used morpholinos to knock out Meis2B, we could see defects in myocardial morphogenesis and defects in the formation of the heart tube in zebrafish. But this was before CRISPR. And so recently, my lab has made a Meis2B knockout, and Didier Stanley's lab also made a Meis2B knockout. And independently, we haven't been able to see any early myocardial defects in those mutants. And there could be different reasons for that. But we decided to test whether, like Pbx factors, whether these Meis factors were also redundant in zebrafish heart development. And so we engineered a Meis1B knockout and crossed that with our Meis2B fish. And we see that zebrafish lacking MEIS1 and 2 also show defects in development of the heart tube. So not as severe as our embryos that are lacking Pbx2 and Pbx4, but we see defects in outflow tract formation and in the assembly of the heart tube. And we think, again, that these are defects in early myocardial differentiation. So we see early myel7 expression as weaker in animals lacking MEIS1 and MEIS2. And so I finally want to end by telling you a recent experiment that we've been doing to further test roles of Pbx and Meis in helping to promote the activity of these important cardiac transcription factors. And one of the factors that we've been testing is Gata5. And so some of you may know that these Gata factors are important and have been used to reprogram cells into cardiomyocytes. Work from several years ago from Jeremy Ryder in Didier Stanier's lab show that if he misexpressed Gata5 mRNA in zebrafish embryos, that this Gata5 overexpression could induce ectopic myel7 expression, indicating that Gata5 overexpression induces extra myocardial differentiation in zebrafish. And so we've thought that we could use this system to ask whether Pbx and Meis are helping to promote the activity of Gata5. And so what we've done recently, so this is kind of a busy experiment, but let me take you through this again. So these are, again, our control animals showing you myel7 expression in the heart tube. If we misexpress Gata5, like was seen earlier, we can see a little bit of extra myel7 expression. If we add Meis or Pbx to Gata5, we see a similar, a little bit of extra myocardial induction. But if we now overexpress Pbx, Meis, and Gata5 together in zebrafish embryos, we now see in some embryos massive induction of myel7 expression. And so this experiment can be, the nature of the experiment is pretty variable from embryo to embryo, but we can pool embryos and quantitate the effects of this induction and show that we can get quantitative increases in myel7 expression by adding all three of these factors together, suggesting that these Pbx and Meis factors are helping to promote the activity of Gata5 and promoting myocardial differentiation. And so, so to conclude, what I've told you about today is a work where we use genome editing in zebrafish to characterize a genetic variant that was previously of unknown significance in heart defects, and we were able to take advantage of looking at early heart morphogenesis defects in our zebrafish and getting large numbers of embryos for those studies. And it's one of the first examples of editing and testing and characterizing a potential human disease variant in the zebrafish. And we think that there will be many more examples in using the fish to do these types of experiments in the future. And then I've also told you about how we're looking at roles for Pbx and Meis factors in heart development. And we think this work is giving us further insight into the transcriptional control of cardiomyocyte differentiation. And we think that some of our approaches can provide a framework for not only looking at new transcription factors, but also other chromatin factors that have been identified as potential candidates in heart defects in humans. Right, so most of the work that I told you about was done by Hank Farr, a research associate in my lab, and assisted by some fabulous undergrads that we've had in the lab, and our collaborators, past and present, and thanks to our funding. So happy to take questions.
Thanks, Lisa. So I think we have time for maybe 2 questions. So raise your hand, and then someone will pass the mic to you.
It's beautiful work. I'm just wondering, is the best strategy, if you're thinking about modeling human disease variants in the zebrafish, is the best strategy to change, homologous variants within the zebrafish in that zebrafish gene, Or would it be to replace that gene with the human isoform and then make the mutations in the human gene in the zebrafish, making sure that the gene functions the same way?
Boy, so actually I hadn't really thought about, so in that case, we would have introduced the human PBX3 gene into the zebrafish PBX4 locus so that it got expressed early, or that we would have, I'm not sure. Essentially, my answer to your question is I think it would be a case-by-case basis, depending on the gene that you're studying, the gene family you're studying, and the relationship of the orthologs. It just turns out that with these PBX genes, even though zebrafish PBX3 is the ortholog of the human PBX3 gene, it's known from zebrafish and mice and humans that the same PBX genes are expressed differently. So for example, PBX4 in zebrafish has important roles in early embryonic development, which is more similar to the PBX1 gene in mouse. So in our PBX gene family, it was I sort of think of the term functional ortholog, although I've been told that that's not really an appropriate way to think about orthology. But so that's my long answer to your question. It would probably be a case-by-case basis.
Yes?
Yes. Do you know where Pbx and Meis bind in the genome? And what are the targets?
Yeah, so there's been lots of work, even in cardiomyocytes, showing that Pbx and Meis binding sites, which are similar but distinct, are present in, say, open chromatin regions and associated nearby with other binding sites for important cardiac factors. And so one thing that we're very interested in now doing in our zebrafish is not only, I think it's important not only to know where Pbx and Meis are binding, but where they're functioning. So now that we've established our mutant lines in the zebrafish, we can ask in cells lacking Pbx, what is happening to chromatin changes and binding of these cardiac factors in the absence of the potential pioneer factors.